The first two videos you learned why spatial transcriptomics matters and how MERFISH works. Now we're going to take that theory into the lab. I'll walk you through the full experimental workflows, explain the reasoning behind each step, and show you what to watch for when you perform these protocols yourself. By the end of this video, you should be able to 1. Describe the main wet lab steps of a MERFISH experiment. 2. Understand the purpose and reasoning for each step. 3. Recognize potential pitfalls and know how to distinguish between good and bad experimental outcomes. I'll also share a practical tip. Because gel handling takes practice and encoding probes are expensive, you can run what I call a practice mode. In practice mode, you leave out the encoding probes entirely and include only the poly-T anchoring probe in the hybridization buffer. This way, you can get comfortable with the gel casting, expansion, re-embedding, and imaging workflow without using up your codebook probes. We start with sectioning, fixation, and permeabilization. Honeybee brains are sectioned to 10 microns on 18 mm cover slips. We dry the OCT about 1 minute per micron and observe RNA integrity and tissue morphology. We fix the tissue in 4% paraformaldehyde for 13 minutes. Then we permeabilize the tissue overnight at 4 degrees Celsius and 70% ethanol. Checkpoint. After fixation, the tissue should look crisp under the microscope with clear boundaries and no swelling or distortion. Weakly fixed samples may show curling or detachment from the cover slip. Next comes encoding probe hybridization, or poly-T only if we're in practice mode. First, we UV treat the humidified chamber, glass plate, and parafilm for 30 to 60 minutes to keep things clean. The tissue is washed in 30% formamide in 2 times SSC for 10 minutes. The hybridization mix contains 30% formamide, 10% dextran sulfate to concentrate the probes, yeast tRNA to block non-specific binding, RNase inhibitor to protect RNA, and poly-T-locked nucleic acid probes that bind to the poly-A tails of RNase. In a full experiment, we add the encoding probes here. In practice mode, we leave them out. We place a droplet of hybridization mix on parafilm, put the cover slip tissue side down, and incubate for 36 to 48 hours at 37 degrees Celsius in a humidified chamber. Checkpoint. After hybridization, the tissue should remain firmly attached to the cover slip with no lifting or tearing. After hybridization, we remove non-specific binding. The sample is washed 3 times in 2 times SSC, then incubated in 30% formamide at 47 degrees Celsius for 2 hours, changing the buffer after 1 hour. This high-stringency wash removes weak off-target probes while keeping the specific ones bound. Checkpoint. The tissue should remain intact after warm formamide washes. Watch for fraying edges or detachment, which can indicate overly harsh handling. Now we embed the tissue into an expandable gel. The monomer solution contains sodium acrylate, acrylamide, bisacrylamide, salt, and buffer. We add fiducial beads for drift correction during imaging, along with TEMET and APS to initiate polymerization. The tissue is incubated in the monomer for 10 minutes, dried, then placed tissue side down onto a droplet of monomer solution. We polymerize for about 2 hours at room temperature, keeping the gel protected from light. Checkpoint. After polymerization, the gel should be fully set, transparent, and firmly holding the tissue. Cloudiness or incomplete gelation can lead to uneven expansion later. Next is digestion or clearing. We incubate the gel in digestion buffer with protein SK for 36 to 48 hours, changing the buffer halfway through. This removes proteins and lipids, making the sample transparent and improving expansion uniformity. Checkpoint. The gel should become optically clear and the tissue should still be anchored without shifting or floating. Cloudy spots may indicate incomplete digestion. With the sample cleared, we move to gel expansion. Before removing the digestion buffer, a siliconized cover slip is placed underneath the gel to prepare for the next polymerization step. Once the cover slip is in place, the digestion buffer is removed. The gel is then incubated in 1 times SSC containing a small amount of protein SK, with the buffer replaced every 20 minutes for 1 hour. During this process, the gel expands about 8-fold in all dimensions, physically increasing the distance between molecules so that fluorescent spots can be resolved more easily under the microscope. Checkpoint: measure the gel's expansion factor. Expansion should be uniform in all directions. Any warping, uneven edges, or irregular shapes may indicate mechanical stress during handling or incomplete clearing during digestion. To stabilize the expanded sample for many rounds of imaging, we re-embed it in a non-expandable gel. This locks the geometry in place. The gel is soaked in the non-expandable monomer, dried, then placed gel side down on a droplet of fresh monomer solution with initiators and polymerized for about an hour. The sample can be stored in 2 times SSC with arenase inhibitor at 4 degrees Celsius for up to a week. Checkpoint: the re-embedded gel should be flat and firm. Wrinkles or trapped bubbles can distort spatial coordinates in downstream analysis. We then do nuclear staining, usually with DAPI. This helps us later in segmentation so we can assign RNA molecules to the correct cells. Checkpoint: nuclei should appear bright, round, and evenly stained under the microscope with minimal background fluorescence. If in practice mode the poly A is stained in a petri dish, the samples are not enclosed in a MERFISH flow chamber but are instead capped on a glass plate with a drop of imaging buffer containing an oxygen scavenger. Otherwise, we proceed to the readout cycles, the heart of MERFISH decoding. For each cycle, we hybridize fluorescent readout probes that bind to the readout sequences on the encoding probe. We then image the sample in imaging buffer containing oxygen scavengers and triplet quenchers to improve fluorescent stability. After imaging, we strip the probes using formamide or cleave the fluorophores with cleavage buffer and repeat the process for the next bit in the barcode. The pattern of on and off signals across all rounds reveals which gene each spot represents. Fiducial beads allow us to correct for stage drift, and clean stripping is essential to avoid false positives in subsequent rounds. Checkpoint: after each stripping step, residual fluorescence should be minimal. During imaging, signal-to-noise should remain consistent across cycles, and fiducial should appear stable in position. Congratulations on learning the MERFISH experimental workflow. So MERFISH is an evolving field, so keep up to date with the latest protocols and improvements to get the most out of your experiments. In the next video, I'll show you some examples of MERFISH data analysis, from reconstructed honeybee brains to spatial gene expression maps, so you can see how the raw images transform into biological insight.